Melania Munoz-Garcia is a biologist with 11 years of experience in the implementation of Nagoya Protocol. As the former ABS national focal point from Costa Rica, she currently manages the German Nagoya Protocol Hub, supporting researchers navigate ABC legal obligation, and coordinates the GNB Hub network and actively contributes with the ABC Compliance Officers Network in Germany. She has been engaging in CBD and Nagoya Protocol negotiation since COP30 in Cancun, first as a Costa Rican delegate and then as member of the DSI Scientific Network. Her expertise bridges science and policy, first in compliance and understanding of ABS framework. We are so happy to have you here and for her interdisciplinary view. And so please, Melania, and enjoy. Okay.
Yeah. Yes. No, the best is to develop the slide to move the slides from the computer and the pointer, the red pointer to the screen. Would be better to switch off this. The light? Yeah, I will put the next right. Switch off the lights. This light.
Don't fall asleep. No. Okay, so then we start. Thank you for the introduction. I work in a culture collection in Germany, the German Culture Collection and Cell Culture. This is a Leibniz Institute, and it's a very important collection. We have more or less 80% of all bacteria that has been described around the world, and we distribute and exchange the strains. So that's why the international regulations for exchanging and using of strains are really important for us. And we are the first culture collection registered according to the European Union Access and Benefit Sharing Regulation. That means that in our catalog, we have all the information related to the restrictions to the Nagoya Protocol. So to start, I would like to say that we want to have today a very interactive session. It's not just listening, Melania. And because of that, we send you a homework, 26 minutes homework. And I hope all of you have watched the video that I shared with you. It has very basic and informative information. And then today we can focus on more practical stuff. As Elena said, I work in the German Nagoya Protocol Hub project, and we help researchers to understand their obligations with the Nagoya Protocol. But it's important to mention that the information we share with researchers is not cannot be considered legal advice. So this is just like tips and recommendations and so on, but you cannot tell the authorities, it's because the German Nagoya Protocol Hub told me that this is out of the scope of this regulation, then you don't have a proof. So it is just, please take it as a good starting point, but it's not legally binding, let's say. So today we will have a super quick overview with some questions related to the video I sent. The video is about the Nagoya Protocol basic information and importance for culture collections. Then we will go through the ABS clearinghouse, that is the main source of information if you are using genetic resources from other countries. And then we will go a bit deeper into the European regulation. I learned that most of you are working in European countries, so I will explain some utilization case studies that are the ones that are within the scope. So first, a quick overview. I would like to ask to you, this is just to warm up, if some of you can say, what is the Nagoya? What is that? What are we talking about today? Sorry?
Come on.
Well, it's just one document, but yeah, it is related to a lot of documents. But what are those documents about?
Come on, it would be very nice if you really interact. It's a good moment. It's among us.
Just get.
It's an interactive presentation, so please wake up.
I will walk around and I will choose a volunteer.
Come on. Bye, Andy. Andy.
Well, it regulates the exchange of sample and genetic information.
Yeah, okay.
International.
Yeah.
Another guest?
You want to add
Something very exotic from the bit of the ABS, the assessment, benefit, and I don't remember the S. Sharing, thank you.
Great. Yeah, so the Nagoya Protocol is an international agreement that regulates access to genetic resources and benefit sharing. And this is under the umbrella of the Convention of Biological Diversity. These all like United Nations regulations that we all should take care about. And then, yeah, it is related to access to genetic resources and fair and equitable sharing of benefits. That means all people using genetic resources should share some benefits back to the countries where you obtain these resources. And it entered into force in 2014. So this date is really important. And then what is ABS? I already said that. What is ABS about? Access and benefit sharing. Just guess very quickly.
Come on.
Joe, what is you should make it ParAqua-COST.
ABS is an acronym that we will be hearing all the time when we talk about the Nagoya Protocol. So it comes from access and benefit sharing. And this is basically that you cannot use genetic resources as you want because countries have sovereign rights over their genetic resources. So it is important.
I think we don't want to know because we don't want to respect somehow. We want to ignore.
It's better to know because if not,
no, I know.
In the future you may have legal consequences.
Yeah.
Then it is important that you must know that if you want to use genetic resources from different countries, sometimes or most of the times you have to ask for a permission first. And this permission will include a negotiation about the benefits you will share with that. So, and these benefits may be things that you are actually doing during your research. It may be share the research results. You may share the scientific publications. You may share different kind of reports, information the country needs to understand their own biodiversity, how to manage their biodiversity, and so on. It could also be a joint international collaborations and also could be training and so on. So we are here talking about training, about how we can collaborate with researchers in other countries. So this all could be counted as benefits that you can share with countries. The thing is that now you need a piece of paper saying that you are committed to share this kind of benefits. And then the question is, if we are talking about...
Andy has the question.
Yes.
To the previous slide, and what you said on the previous slide,
Yeah.
Sharing and registering this sharing process. We had international projects, and then we asked about in the Nagoya office that we have at the Federal Office for the Environment. And they were quite simple, the processes that they obliged us to do. I don't know if this was particularly for Switzerland, where usually we do things in a complicated way, but there I was surprised to see that it was surprisingly simple.
Mhm.
They said, well, if it's a research collaboration, you already have most of this information that you would need to register, like maybe you're on a proposal together and there's described, these are two partners, international partners that work on a project, the type of sample that you will generate from the project or genetic data, and so on. And because it's not for commercial research, she said that information that you have there, you design a project, work packages, that this is basically what you need.
Yeah, this is
And also
important.
from where you're at.
This is interesting because the important thing is you should know where this biological material originally come from,
Yes, ma'am.
right? So, for example, if you are taking a biological material from Brazil, then you have to check the Brazilian legislation. If you are taking biological material from Colombia, you have to check the Colombian legislation. From Spain, the Spanish legislation. And then when you comply with the legislation of the samples, then you need to check within your European country what are the compliance regulations, because what European authorities are doing is checking that all researchers within their countries comply with the laws from the other countries. So, for example, in Germany, we have user checks. That means the authorities in Germany knock the doors of the research institutions and ask, please give me a list of the projects that are within the scope of the Nagoya Protocol and give me all the necessary permits. Then researchers have to collaborate with that. And if not, they could be subject of fines and stop research and other kind of legal consequences. That's why it is important to be informed and to be aware of this, because it is better to work on this with time that suddenly the authorities knock the door and ask you for all the papers and you don't have them. And then it's going to be a very important rush there to obtain everything.
Yeah,
Yeah.
Even though like other countries out
Can you please raise your voice just for people home?
So even though that some countries out of Europe is part of Nagoya Protocol agreement, we need to check both sides if they reflect their sources.
Yeah, if your samples come from countries within Europe or outside Europe, because the access laws are not regulated at the European level, but are regulated at each member state level. So, for example, some countries as Germany or the Netherlands decided to do not regulate access to genetic resources, so you can use genetic resources from Germany or the Netherlands or Poland with no permits. But on the other hand, Spain, France, Crete, Croatia, and other countries decided to regulate. So you have to check with those countries. The same that you have to check with Latin American or Asian countries. I will do one thing because I am a bit worried about timing. So let's say that if you have questions related to the video at this point, we will answer those questions. And then if you have more specific questions about the specific projects, we answer those at the end, okay? We will have time. But then, if we are talking about genetic resources and derivatives, can you tell me what is a genetic resource? Yeah, if you have to know if you are using genetic resources or not, what is a genetic resource? What are we talking about?
DNA sample.
Who answered?
Me.
Yeah.
DNA sample or
DNA samples?
DNA can be extracted from.
Yeah, perfect. So genetic resources is all related to biological material that contains DNA and RNA. Easy, right? So it could be animals, plants, microorganisms, whatever that is not human, that contains units of heredity, that means DNA or RNA, right? And the other concept that is a bit more complicated is derivatives, because scientists don't use this concept. This was invented by the United Nations, so it is a bit more complicated, but it's basically biochemical compounds. So when you hear derivatives, it's all biochemical compounds. That means if you are studying metabolites or proteins or enzymes or lipids or biochemical reactions or fermentation reactions or also, I don't know, maybe you are working more with microbes, but if you are studying honey and milk and wax and other kind of products of the metabolism, you may be within the scope of the Nagoya Protocol too. Yeah.
I have a formaldehyde processed specimen from which I cannot extract DNA. This is how biological.
If you cannot extract DNA,
because it's impossible from formaldehyde fixed.
yeah, it's not at genetic resources. So if you are, for example, we'll go to utilization later, but if you can extract DNA, this is a genetic resource. So people sometimes study archaeological samples, and they are still able to extract DNA from archaeological samples. So this is considered a genetic resource. If you are studying herbarium samples, so even though they are dry, you may be able to extract DNA. So it is considered a genetic resource. Of course, if you are studying living microorganisms, of course, they are considered genetic resources. So all of you are working with genetic resources, microbes. And then I would like to explain very quickly the three pillars. Access is the permit you need to obtain from countries, and this is regulated by each individual country, even though this country belongs to the European Union. Then you have to sign a contract and make commitments and say, I will share these benefits with you, country, provider country. And then the compliance is the one that is regulated at the European level. So that means that there is an European law controlling that everybody in the European countries access genetic resources according to the national legislation in the provided country. Yes.
Yeah, I want to ask. So basically, the Nagoya Protocol then is regulating the circulation of samples, so biological material, right? But in the agreement, the benefit sharing, if I understand correctly, means that this is about also the data that are produced.
Yes, it's both. But I have to clarify that just moving or holding genetic resources is out of the scope of the Nagoya Protocol. What is within the scope of the Nagoya Protocol is the research you do with that. And I will move to the next slide that is related to what collections do and what is this important for collections, right? So holding genetic resources and transferring genetic resources is out of the scope of the Nagoya Protocol. If you receive these genetic resources and you do research with these genetic resources, then the responsibility to request all the permits is in the researcher receiving the resources and utilizing these resources. So even though you obtain a strain from an ex situ collection, you have to check. I mean, let's say the ex situ collection is here in Italy. So you go to an Italian ex situ collection and you take a strain you are interested in, and then you go to Germany and you want to study that strain. So what you have to do, you need to ask to the collection, which this strain was originally collected. So they tell you, well, this strain is from Brazil and it was collected 4 years ago. And then you say, okay, then even though you obtain the strain in an Italian collection, Italy is not the provider country. The provider country is Brazil because it was originally collected there. And then the date is important because, as I said, the Nagoya Protocol entered into force in 2014. And all the samples collected after 2014 are within the scope of the regulation here in the European Union. The difference is that, for example, some countries have the ABS legislation even before 2014, and you may still comply with those laws. I will explain that a bit deeper, but you need to find out where this material was originally collected and when, and then you have to check according to their national laws if you have legal obligations. The tricky part is when you obtain just one strain from just one country, you have to understand just one legislation and follow one procedure. But if you want to obtain 20 strains from 10 different countries, then it gets a bit more complicated. So it is important, and that's why I included here, that is a good practice for biobanks and ex situ collections in general, to have all the relevant Nagoya Protocol information available. And this was explained before. If the people obtaining these strains will need a permit to use them. This is what collections should have. Where this sample was obtained and when, and according to these 2 points, if you have obligations on the Nagoya. As I said, this is not an obligation under the European law, but it's a good practice, let's say, for collections. I don't know if with that I answered your question or I.
The data.
Oh yeah, the data is true.
There are obligations related to that, and if there are possible.
Yes, yeah. So if you are doing research, and this research involves genetic resources or biochemical compounds, then you have the obligation to obtain your permit. And then the publications and the data you publish is one kind of benefit sharing you can share. But of course, if you are doing research, you are producing new information, you're producing data, and this data is, as I said, a result of the research, but you need to obtain your permits before to start your research. And related to DNA and RNA sequences, this is another a bit complicated topic that I won't go in deep today, but there are still important negotiations about benefit sharing about these sequences, because now researchers don't need to go to a collection to obtain a strain, the material, the living microorganism, but you can easily obtain DNA and RNA information from websites. And then there was a huge negotiation because countries considered this a loophole in the Nagoya Protocol, because the researchers around the world and the industry around the world is using the DNA sequences from databases to develop commercial products. So it was a huge negotiation because countries wanted to have a bilateral system. That means you need to ask individual permits to individual countries to use sequences, but this is basically impossible, right? In our day-to-day life, because you use hundreds or thousands or millions of sequences to perform just one analysis. So to make the story short, at the end, what was decided is to have a multilateral mechanism for benefit sharing. So you don't need to ask individual permits. That's good. And then there will be a fund where companies that commercialize products coming from genetic resources will share monetary or money with the fund, and the fund will distribute this within the country. The good thing is that for basic research, you don't have any obligations. Then researchers can continue using genetic sequences as usual. So you don't have to worry about obtaining sequences from the databases, and you are still free to do that.
Sorry, just before we move on, what happens in a lens of no one? So Antarctica, for instance, that is not belonging to one country. So who, or more or less, who should we ask for permissions for? I don't know. Antarctica, for instance, or
if you once again, if you
Antarctica.
Antarctica, that are not belonging to just one country.
Yes.
So how do we?
Antarctica is out of the scope because the Nagoya Protocol has the scope just within the jurisdictions of countries. So areas like the deep sea is out of the jurisdictions of countries. The Antarctica is out of the jurisdiction of countries. So it's out of scope.
Okay. Yeah, but sooner or later, every place will be. So it's really important to enter into this mindset that you have to know everything about, because at a certain point there will come something that we say starting from 2025.
Yeah, so
That's
for
what
example, 2 years ago, the BB&J agreement was adopted. So the BB&J is the biodiversity beyond national jurisdictions. And this is regulating everything in marine waters that are not belonging to any country, the deep sea and so on and so on. It's regulating minery and some other things, but it also has a chapter about genetic resources. So the BB&J agreement is not in force. They need the ratification of 50 countries, and maybe it entered into force in 2 years. I don't know. It could be in 1 year, it could be in 5 years. It depending how fast the countries ratify this agreement. But this is coming. So in the future, we will have access and benefit sharing rules also for the international waters, but not belonging to the Nagoya Protocol, but belonging to this other agreement. Thank you.
Yeah, because Antarctica, not whole, but some part, which have special agreement between is the Antarctic Special Protected Area.
Yeah.
So this is only one part, this is special agreement, but the rest is.
Yeah, correct. Mm-hmm. And then another important point, as was discussed in the talk before mine, is that when you describe new species of microorganisms, this is really important because when you describe new species, you characterize the species, you describe the functions of the genes, you describe a lot of things, and this may be within the scope of the Nagoya Protocol, but a lot of parties or a lot of countries, they prohibit the distribution of these strains to third parties. So basically, if you deposit a strain from specific countries, at this point, I remember Brazil, India, and there is another one, that collections are not accepting the strains, new species of microorganisms in these collections because the restrictions related to the Nagoya Protocol. So this is very relevant for us, because then you need to take care of that, because if you are describing a species from a country that established restrictions, then you will not be able to publish these new species because the collections will not accept your type vouchers. So take care of that.
What about them is that when I buy it from a commercial collection and the organism is from another country,
That's what,
do I need to do something? Or is
that's what she
Yeah.
said.
You need to check which is this other country and check if this other country has regulations. And then you have to follow the regulations from that other country.
We come back to get back on this because we already answered, but we go on a little bit and then we come back, okay? To this
I
point.
will be available during lunch and getting coffee, so
We will have Melania the whole day. We can squeeze with questions.
I will skip the questions here because you have asked the questions, and then I will move to the practical thing. So ABS asking for permit and sharing benefits, it sounds easy, but it is complex because every country do this different. And for us working in the European Union, when we talk about the Nagoya Protocol, we need to have 2 different compartments in our heads. The first compartment is related to the obligations in the provider countries. And they do ABS differently. So basically, you need to identify the provider country and check and understand the law in that country and see if you have obligations. We have this interactive infographic, and we have some questions that will help you to identify if the law applies to you or not. This is compartment 1, and compartment 2 are your obligations under the European Union ABS regulation, and this is more about showing compliance. So showing compliance means go to your country authority, to Spain, to Poland, to Switzerland, to wherever you are doing research, and show them, I have my permits, I'm compliant. And for that, the European Union has this declare portal where you make a user, or your institution has a user, and you can declare there. This is an obligation under the EU. You must go proactively and declare you have your permits. Okay, So box 1, box 2. And I say box 1, box 2 because the European regulation has a specific scope where the authorities will check on you, and the provider countries could have a broader scope. So it's tricky. Sometimes you are out of the scope of the European Union, but you are within the scope of the law in the provider country. So that means that the European Union will not check on you because you are out of the scope of the European regulation. But if somehow, for example, Peru, if you access the genetic resources from Peru, Peru has a commission checking scientific papers and patents and so on, and they are checking if someone is using Peruvian genetic resources and they don't have the permits, so they can catch you. So you may have responsibilities with the provider. Okay, so the clearinghouse. We discussed about this. The first step is you have to check where the material comes from, which is this originally, when the material was originally collected, not where the collection is, but where the material was obtained original. And you need to check if they have storage.
Daniel is like, oh, this cannot be.
So here we go to the ABS clearinghouse. So according to the survey, few, just like one or two of you have experience with the ABS clearinghouse, and that's why I ask you to bring your computers. So the ones that have computers, you go to this web app. To AB is www.absch. Or maybe I should have, well, I will give you the website.
We can put it in.
No, I have it in another slide. No worries.
No problem. So we can find immediately.
So the point is that the ABS clearinghouse, you will see a map like this, and then you will see which are parties to the Nagoya Protocol, that is the green countries, and which are not parties, that are the gray countries. So you can see which countries, and then when you click on top of one country, you will see the country profile. And this is very important because here you will have all the information you need to understand. So you will have what is the contact people that is working at the government, what is the legislation, what is the procedure, if they have grant permits. This is the international recognized certification of compliance. So as you can see, France have granted a lot of permits. So for sure, if a country has granted permits, they regulate access. Then you can access national websites, and then you can check the interim report. The interim report is sometimes useful when a country hasn't published anything in the clearinghouse, because sometimes you find a lot of information and sometimes you find empty profiles. So the empty profiles are the problem. But what I want to say is that this website is the most important source of information you can find to check that the legislation in the provided countries.
I like the Italy. It's great.
Italy, I have to say, Italy is great. Italy as a country is not party to the Nagoya Protocol, but the European Union as a whole is party to the Nagoya Protocol. And then even though Italy is not party, you have compliance obligations.
No, but we are in the Court of Justice for this. So Italy has been sued by Europe because we didn't implement because as a general thing, directive, European and whatever directive is we must compliance with this. So we didn't. So that's why we are under the Court of Justice also for this.
And a platform about compliance research about genetic resources, but like all findings about European regulations that
Okay. So this is the key information. For example, if you have seen that the country has ABS procedures, that means that they regulate. And if they have granted permits, that means that they regulate. So this is the website. So ABS-CH, CBD for Convention of Biological Diversity, and INT. And you can have it in English or in the UN languages. So now the ones that have computers, I will ask you to open this website. And the ones with no computers, I will ask you to sit next to one who has a computer. It's because the mobile phone version is not that nice. So you cannot find the map. And the other thing I will ask you is you scan the QR code here. I don't know if your phones comes till here. Or the other thing is go to menti.com, and include this code, 86478088. So you have to check the ABS clearinghouse in your computer, and in your mobile phones, try to access the QR code. Let me know.
Or something like this, because I don't know if you ever use this Mentimeter, but it's an interactive platform. She can answer, and we can immediately visualize the answers. So
Yeah, I will ask you some questions, and then you can answer through your phone.
let's see how
And we will works.
Yes. Sorry.
Everybody in the Mentimeter, raise your hands. Who is already Mentimeter? Okay. You ready in Mentimeter?
Trying to connect this.
Okay, but important also to, did you connect to the clearinghouse? Then you maybe can share, but the most important thing is you're connected to Mentimeter, then you can participate in the quiz. So exploring the clearinghouse is a very important key thing. Okay, then we will start. So the thing here is that if you answer fast, you obtain more points. And if you are, this is the game. You have to answer the questions. I have 10 questions through the whole presentation. And then if you answer fast, you obtain more points. And if you answer correctly, of course, you obtain more points. And at the end of the presentation, we will have a score. And we will have the three winners. And for the winners, I have some bags. Cha-cha, with the Nagoya Protocol Hub logo and some pins. And here I have few stuff that you can all use. Okay, but we are ready. Answer fast to get more points. Then, checking the clearinghouse. Is Poland a party to the Nagoya Protocol? Yes or no? You have to click on top of Poland, or you can just see the color. And sometimes, if you don't know where a country is, you can also have a search by country at the top, and you can write the name of the country. Okay, here are the answers. Poland is not a party to the Nagoya Protocol. So when you go to the clearinghouse, you see Poland here, and Poland is gray. So Poland is not party. Just the green countries are party. You have a small legend at the bottom. And then, as I said, if you don't know where a country is, all the, no, but at the top of the clearinghouse, you have country profiles. If you check the clearinghouse, if you go to the top, you have in the menus, you have the country profile, and you can write the name of the country, and you will find the country. Okay, next question. Does Argentina regulate access to genetic resources? Yes or no? Or do you think it's not clear in the clearinghouse? I added this because sometimes you don't have enough information in the clearinghouse to know. So go to Argentina, open the country profile, check if they have granted international recognized.
It's great.
You're getting better. You're getting better.
Argentina is not great. It's great. But pay attention to the question. I'm not asking if Argentina. Silence. I'm not asking if Argentina is a party. I'm asking if Argentina regulates access to genetic resources. This is a different question because there are parties that do not regulate access to genetic resources. For example, Germany is a party to the Nagoya Protocol, but Germany do not regulate access to genetic resources. So if you go to Argentina, well, before you click on top of the name of the country, Argentina is green. So Argentina is a party to the Nagoya Protocol. And then they have ABS procedures, and they have granted internationally recognized certifications of compliance. That means Argentina has granted permits before. That means they regulate access. Okay. Then you have legal obligations with Argentina if you are using strains from Argentina. Next question. Does Montenegro regulate access to their genetic resources? So if you don't know where Montenegro is, go to the top of the menus, click on the country profiles, write down Montenegro. You can see
It's too small, I cannot reach it.
You can zoom in,
I only see.
So remember, I asked you.
Was too fast.
Pay attention. Yeah, you have the 60 minutes because we have to finish this presentation at some point. So pressure, there is pressure. Okay, just one person answered correctly. When you check, pay attention. When you check Montenegro, and you check the key things I told you, have they ABS procedures? You don't know. Because this is very important. When you have 0, it doesn't mean that they don't regulate or that they don't have ABS procedures or they don't have anything of that. It may mean that they haven't published their information in the clearinghouse. And this is a big problem for many countries. So when you find a profile like this with a lot of zeros, you cannot trust. This is the most important message here. The ABS clearinghouse is a very important source of information, but when you have 0, you cannot trust. You must ask the question to these countries' authorities. So if they have 0 legislation, they may have legislation, but they haven't published this in the clearinghouse. And this is a very important campaign we are doing, trying to convince countries that users need this information to comply, because if they don't publish the information in the clearinghouse, it makes everything much more complicated. So, key message, if you have 0, you cannot trust. What you have to do in this case, you have to send an email to the ABS national focal point or the competent national authority. And you can ask, I have a research project, I am doing this and this and this. I would like to access or to utilize material from Montenegro. Do you have any regulation that I have to follow? And if yes, send me the procedure, send me what I have to do, send me the forms, send me the information. Yeah.
I was going to ask a question. So is it important to always have these exchanges in written form?
Yes.
Yes. And I'll explain why. Because, for instance, one of the projects we have, I talked to, so I sent an email to ask a few questions, but the person actually called me and we had like a 1-hour phone call where the person was asking me many things. And he said, yeah, so according to what you tell me, you are okay, you can proceed, everything is fine. I do not have a written response from them because I assume because they called me, everything was okay. This means it's not.
Yes, it means it's not. And this is a recommendation. It is good to have a phone call or a video call. This is very helpful because you can exchange all the details. It is much faster than exchanging 20 emails. But at the end of the call, you will say, okay, I will just send you an email that you confirm what we have discussed today. Then you send an email to the authorities saying, dear blah, 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 I am sending this email after our video call or phone call. just to confirm that for my project, title of the project, we don't have any ABS obligations. Please confirm. And then they answer, yes, you don't have obligations, and then you have a written proof.
Okay.
Why? Because according to the EU regulation, you need to show compliance. And then if the authorities knock your door and you tell them, and they ask you, where are your permit? And you tell them, I don't need a permit, and they ask you why, then you can show the email.
For 20 years, no? So you need to keep the number.
Yeah, keeping the emails if you don't have obligations.
20 years.
Keeping the emails if you don't have obligations is not an obligation according to the law to keep them, but it's useful. It's a recommendation from our project. Keeping the permits, when you have obligations, you obtain your permits, you declare that you will have your permits, then you have to keep these permits for 20 years after you finish your project. And this is in the law. And the most important part is that the institution should have a way to keep these permits, because researchers and students come and go. Then if you have visitors, if you have PhD students, if you have staff that is not fixed, then it is important. Clear? Let's move to the next one.
Oh, no.
Then, if you have to write an email to the national focal point in Montenegro, who is the national focal point in Montenegro? Is Tamara Brejovic, is the Ministry of the Environment, or is not clear in the clearinghouse?
Yeah.
Who is the national focal point in Montenegro? Yeah, it's Tamara. So, when you go to the Montenegro profile and you click on top of the national focal point, you will obtain the name of the national focal point.
I will remember Montenegro forever. After this exercise. I want to go to Montenegro now to check the example.
And then if you click on top of this, you will find the email address. So if you need to write an email to Montenegro and ask what you have to do, go to the country profile, go to the national focal point. I recommend to send an email to everybody, to the national focal point, and to the competent national authorities. Somebody will answer your question. And be patient and be insistent. If you haven't received an answer in 2 weeks, send another email. And if you haven't received an answer, send another email, because you, according to the EU ABS regulation, you must do your best effort to find if you have obligations with.
Take a flight, go to the Montenegro first.
Yeah.
Take the first.
Tell them.
Silent consensus, no.
No, it's not
Not
valid.
valid.
So you must do your best effort, but they don't describe what is your best effort. So if you show them you have sent 5 or 10 emails, then maybe they consider you did your best effort. Okay.
Be careful.
Then the last question is, does Germany regulate access to genetic resources? Yes, no, or it's not clear in the clearinghouse?
No.
Remember, not asking if it is a party.
Mistake.
Remember, I'm not asking if Germany is a party. Parties can or cannot.
Incorrect.
regulate.
No, yes. I'm trying to.
So, pay attention. Germany is a party. Germany too is a party of the Nagoya Protocol. But Germany does not regulate access to the genetic resource.
Yeah.
And I included this example because then I will dive you into this. Okay? So you can see party to the Nagoya Protocol. Okay, Germany is a party. Then they don't have ABS procedures. guys, they don't have ABS procedures and they haven't granted any permit. So you say, okay, maybe they regulate or maybe not. And then if you click on the legislative or administrative procedures, the ABS laws, you will see this. It's a lot of text. But basically, if you click on your computers, the text of the ABS legislative procedures, you will see the laws of the European Union. And remember, the laws of the European Union are not regulating access to genetic resources. They regulate compliance. So it's different. Germany is not obliged to regulate access according to the EU. Germany can decide if they regulate or not. And if you scroll down in here, you will find that they have no provisions for access and no provisions for benefit sharing. That means they do not regulate access. And I will explain this again. They do not regulate access. It means for you that you don't have to request any ABS permit to Germany if you are using German strains. Then, if you go to the Italian collection, you obtain strains that were isolated in Germany and bring them to Italy, you don't have any obligation because Germany doesn't regulate access. This is what every global member state can decide if they regulate. So Germany doesn't regulate access. What Germany is obliged to do is to implement the EU regulation on compliance. And Germany does that. Germany has an authority that knocks the door to the scientific research institutions and companies and asks for the permits. So they do check compliance and they do not regulate access. Is that the difference there?
Yes.
Yes.
But I don't know if for everybody's clear.
Okay. And then one other thing is the interim report. So if you go to Germany's country profile and you go to the last thing, that is the interim report, and this is important for all countries, you can check question number 11. Write it down. Question number 11 in the interim report, you cannot see them from there, but you maybe can see them from your computers, is asking if access to genetic resources is subject to PIC. And PIC is the permit. Okay? And Germany says, no, there is not a specific legislation on access to genetic resources as well as benefit sharing in Germany. Accordingly, there are not requirements to obtain PIC and MAC, that means the permit, when accessing genetic resources in Germany.
What is the PIC and MAC?
The PIC and MAC is... No, the PIC and MAC is another acronym. The Nagoya protocol is full of acronyms. The PIC and MAC means the previous informed consent and the mutually agreed terms. And this is basically a complicated way to say an ABS term. But when you see PIC and MAC, it is related to the ABS permit. Okay, so you feel more comfortable now navigating the ABS clearinghouse?
Yes, apart from the study geography.
And understanding how to interpret the data. The most important thing is you find zero information asked to the country. Okay? And this is a general overview of what European countries don't regulate access, the one in green, so you can use genetic resources from these countries with no permit. Then the one in yellow are countries that regulate some kind of genetic resources, but not everything. For example, in Portugal, you don't need a permit if you obtain strains from mainland Portugal, but if you go to the islands, so Madeiras and the other one, I forgot the name, Azores, they do regulate access. So you need a permit just if you obtain strains from those islands. And the ones in red, they do regulate access to almost everything. So Spain, France, Greece, Malta, other countries, go and ask and request your permits. And keep in mind, this is not legally binding. This is just the research we have done. Things can change. So then the country can decide to regulate and establish new legislation. And the countries that are in gray is because we are not sure if they regulate or not. So we need to do a bit more research. Then I think we are out of time, right? So we want to have the time to do the other part of the quiz.
I think we can catch up in lunch break.
If everybody agree, we can go on. How much?
I think there is a break now.
Yeah, but we can regulate. I mean, if everybody is interested and
Maybe 15 more minutes and then drink the
15 more minutes are okay for you? 15.
And then you can drink the coffee and the cookies and eat here while the other things.
Yeah, it's fine.
Yes? Will you survive? Okay. Then, as I said, you have obligations with provider countries, and this is included in the infographic, and you can check it later. And we are developing these easy ABS guides. This is just an announcement that you can check easy to understand information about ATS in these countries in our website. I will share with you the presentations with the link. But then when you enter into the European Union, you need the authorities here will check you on compliance. And then you are in the scope of the EU regulation. All this is in the presentation, so I won't go deeper. But if you answer yes to these 4 questions, you are within the scope of the EU and you need to show compliance here in the EU. And it is important to mention that those are accumulative. So if you have no as an answer in just 1 question, you are out of the scope. So you are free to do.
That's nice.
So you need to answer yes to all of them to be within the scope. If one is no, you are out.
Generally, it's the opposite. If you say no, but you are yes.
And the most important question here, or not the most important, the most tricky one, if you are doing research on the genetic or biochemical composition, that this is what is within the scope. So utilization, the European regulation has a definition of utilization, and they will check you if you are utilizing genetic resources. So, and this is research on the composition of the biological material. So if you are doing this kind of research, gene function, expression, modification, genome editing, all these methods are within the scope. But if you do just storing biological materials or just culturing, even though if it is in vitro in a lab, if you are not extracting DNA, if you are not extracting biochemical compounds, you are out of the scope. If you just use 16X or some kind of barcoding to identify your strains, this is not considered utilization under the EU. So once again, European authorities won't check on this, but you still may have obligations with the compliant and with the provider countries. Keep in mind your 2 boxes in your brain. And then morphological and anatomical characterization is out of scope because you are not doing research on the DNA. Then some examples. We will practice this. You have still the Mentimeter open, and then remember that all cases included here are related to the compliance part in the EU, but you still may have obligations with the provider countries. Okay. And this is a very important document that I will recommend you to check. It is a bit long, but it is very informative. It has a lot of chapters that you can focus on your chapter you are related to, and it gives a lot of examples. And reading the examples, you will understand what utilization means. So this is my huge recommendation today.
Thank you.
Read the guidance document. Then go to Mentimeter. You don't need the ABS clearinghouse anymore. And then we will check if this research is considered utilization or not. So the storage of pathogens pending a decision on their use in a vaccine. So you have a collection, and different kind of pathogens are isolated from different countries as part of a global survey. So they will be considered from epidemiological analysis to be potential public health threat. So you are not doing any analysis at this moment. You just isolate and save them. And then in the future, at any moment, they could be used for a vaccine development, but not yet. So these are just stored in your collection. Is this utilization according to the EU regulation? Yes or no? So basically, if this, you have a collection, you are keeping these pathogens could be used in the future to develop vaccines in case of a health emergency, but not yet. Is utilization or not? Do you have obligations or not? Correct. This is not utilization because you are just storing the material. You are not doing any research on this yet. So this is the answer. It's out of the scope because you just have a collection and you're not doing research. But in a later stage, if you choose some vaccine candidates and you start the research to develop these vaccines, you cross the line and you enter into the scope of the EU regulation and you need to show compliance. You need to show your permits. Okay, case number 2, taxonomic identification. So the study involves taxonomic identification by comparisons with different sequences, 16S or barcoding of a pathogen to determine with which disease a plant has been infected. So is this considered utilization? So basically you go and you obtain samples from plants, you extract all the microbes there, and you want to know if this plant is infected with a disease caused by a microbe. Is this utilization? Okay. At this moment, it is not utilization. Why? Because according to the EU regulation, just taxonomic identification, even though you use DNA, even though you use 16X or barcoding, you are not studying the genetic composition. You are not describing characteristics. You are just comparing and you're not discovering anything because the 16S is there and you just make a blast or whatever and you just identify the species. This is out of the scope of the EU regulation and the EU authorities won't check on you. But you may still have obligations with the providing countries because most of the countries regulate taxonomy. So even though they will not check you, the provider countries may check you.
Just one question, because here is a comparison with a reference sequence. What if it is a new sequence, a new fish, or a species that has been described, but it's the first time that this sequence is being, for the barcode, that is being produced?
If you're describing a new species, then this is not taxonomic identification per se, because taxonomic identification is, for example, if you collect soil samples or water samples and you want to identify which species are there. But if you are describing a new species, this is another thing. If you are describing a new species and you sequence the whole genome and you want to deposit this and you want to describe the new species with a publication, if you just sequence the genome and upload the genome, this is out of the scope because you are not studying the functions of the genes. But this almost never happened, right? Most of the time you describe the gene's functions and you're saying this new bacteria metabolizes this and this and that. They have these genes producing these enzymes and there is new characteristics, and then you are describing for the first time the functions of that microorganism. So when you go to functions, it is within the scope and you will need permit and you will need it.
If it's just the barcode, so for instance, for fungi, we use ITS barcode, and sometimes we use the barcode to delimitate the species because maybe based in morphology, it's not enough. But sometimes the species has been described in the past. It's just that for some reason, It's the first time we are producing this sequence. It's still false is or out? Out. Okay.
Because barcodes are not
within the scope of.
No. Yeah, barcodes in general are not within the scope because barcodes doesn't have any function. It's just the 15 eggs and so on. So you are not studying the function of any gene or anything because a barcode usually is not a gene. Okay. But if, for example, after you collect the samples and you identify the disease and the species, is out of scope. But then even if in the future you do some genotypic analysis and check phenotypic differences between individual pathogens and strains, and you start researching the virulence and which are the metabolites included or involved in the virulence and so on, then you move into the scope. Okay, let's move. We have 5 more minutes. Case number 3 is the characterization of extracts, different types of active compounds present in a biological control agent. So you extract these biochemical controls, these biochemical compounds from the genetic resource, and then you characterize them and you establish the chemical structure and function of the compounds for the use in biological control. Is this study considered utilization? Then you extract these biochemical compounds, you characterize the compounds, you describe the functions of these compounds to be used as biological control. Is this utilization or not? Yes, it's utilization because you are studying the functions. So basically keep in mind that according to the EU, studying the functions and the new characteristics is important. Then I will show you a couple of relevant cases that has nothing to do with utilization, but about what kind of material is within the scope. So when you have associated organisms of unknown origin, so you go and, well, I will read. A contaminant organism is discovered and isolated from microbial strain deposited in a collection. So you have a collection, you have the cultures, and suddenly you see a contaminant there, and you isolate this contaminant. This contaminant could have originated from the country of origin, where the strain was collected, or from the country where this strain was deposited, or somewhere in the middle. So is this material within the scope of the EU ABS regulation? Remember that according to the EU, you must do your best effort to understand which is the provider country. Then, do you know the provider country of this strain? It just appeared there. It is within the scope. So the answer is the material is within the scope or out of the scope. It is out of the scope. It is basically because you cannot know the country of origin of this material. It could be originated in any country. You cannot test or you cannot prove where this material was originally come from, and then the European Union said, well, if you don't know, then you don't have any compliance.
And you have no possibility to know.
You don't have any possibility to know. I mean, if you import a very sealed vial that you know that this, all the microorganisms involved or included in this very sealed thing, then you have obligations because you know where this material comes from. But a contaminant in the collection is very difficult. Could be someone from somewhere that could bring this contaminant to the collection. A national or international student or whatever.
Zacche di Streptococcus.
Then last example. So pathogenic genetic resources and pests introduced unintentionally to the EU territory. A new viral disease of tomatoes was first observed in the Near East in 2014 and has since been, and from this moment has been detected in the EU. Then the virus isolates taken from the virus isolates were taken from important fruits. and are now under analysis. So if these will be dissolved.
I feel like I'm going to jail.
So for example, you go,
Can I be arrested?
so you work at the Ministry of Agriculture and you're doing inspections and you are checking for pests in farmers markets and you take samples from all the tomatoes there. And then to analyze this disease and to do research with this disease, you have obligation. Yes or no? No, you don't have any obligation with the EU, basically for the same reasons. When you are sampling microorganisms in the farmers market and these tomatoes were imported, you don't know if the microorganisms that are on top of these tomatoes come from Italy to sample here, or come from the boat or the ship, or come from the provider or the original country. You don't know where these microbes come from. So this is how it's called, because you cannot ensure where these come from. This is a bit different if you are studying the tomatoes. If you are studying the DNA tomatoes, you know for sure where these tomatoes come from. You know for sure that the tomatoes come from Italy, or the tomatoes come from Greece, or if the tomatoes come from Costa Rica, because you are importing the tomatoes from somewhere. So if you study the microbes, you don't know where the microbes come from, but if you're studying the tomatoes, then you are within this. And then if this disease, for some reason, you planted the tomatoes here, and this disease that was not in Italy before, now is in Italy, and this is established in Italy, and you collected these microbes in the field in Italy, then you have to follow Italy's ABS laws. As Italy, as long as we know, don't have any ABS laws, then you can use them freely. But if these tomatoes are established in Spain, then you know for sure that Spain has ABS laws, and you must follow ABS, the Spanish ABS laws.